Welcome, students, to the last part of part 1 of chapter 3. So we're going to finish up kind of looking at the plasma membrane. So in class, we talked about passive transport processes, how we can move things into or out of our cells without using energy. Well, we have two types of active transport processes. One of them is just called active transport. It uses things called basically solute pumps, and the other one is vesicular transport, which basically uses little sacs called a vesicle. And we're going to talk about kind of both types. The reason we need active transport processes is because a couple of different things. Maybe the thing that we're trying to move is really, really big. It might be too big to fit through an ion channel. And so, when you're trying to think about moving something heavier, right, that takes more energy. If something is not lipid soluble, then it can't move through the membrane, because remember, the interior part of our plasma membrane is hydrophobic. If something is lipid soluble, it fits in that hydrophobic environment, but if it's not lipid soluble, it's not going to fit in that hydrophobic environment. The last reason we might want an active transport process is if we're trying to move something not kind of downhill, but uphill. So if we're trying to move something like glucose kind of up its concentration gradient into an area where it's already in a pretty high concentration, then we need energy to do that, right? If we're going uphill, that needs energy. And of course, our energy is always in the body of ATP. When we are talking specifically about kind of active transport that isn't vesicular transport, then we are using these things called pumps, a solute pump essentially. What they really are basically carrier proteins embedded in the plasma membrane that can bind onto whatever it is that we're trying to move. They'll change shape and then either kind of let that thing into the cell or release it outside the cell, and then it'll kind of reversibly let that thing go and kind of go back to its original shape. These types of solute pumps are always moving things against their concentration gradients, and so they will require energy in the form of ATP. The most well-known of the solute pumps happens to be this one right here, which is the sodium-potassium pump. So let's look at this here. Let me zoom in. So you can see here is the inside of the cell. Here is the outside of the cell. We have a higher concentration of sodium outside the cell than we do inside the cell. And if we're trying to move sodium out, we're trying to move sodium from a low concentration to a high concentration, and so we're going against the concentration gradient, and so we need energy in the form of ATP. So here's our little solute pump. You can see it has 3 little binding sites for 3 little molecules of sodium. ATP will come and we'll split it, release that energy after our sodium bind onto it. That allows the protein carrier, the little solute pump, to change shape. And then the sodium are released into the exterior of the cell, so into the interstitial fluid. Well, this is a sodium-potassium pump, meaning it moves both sodium and potassium. Well, potassium is higher inside the cell than it is outside the cell, and so we're trying to move potassium also up its concentration gradient. And so again, we use this little solute pump. The potassium come here and bind on to the little pump. We release the inorganic phosphate, the last little bit of our energy from our ATP. That allows the carrier protein to change shape again, and that allows then the potassium to be released into the inside of the cell. Another molecule of ATP binds onto our little carrier protein, and it's ready to do it all over again and pump 3 sodium out and 2 potassium in against their concentration gradients. That keeps a very high concentration of sodium outside the cell and a very high concentration of potassium inside the cell, which is vital for the functioning of our nervous and muscle systems, as we'll see a little bit later in the semester. But pretty much every cell in your body has boatloads of these sodium-potassium pumps. Let me zoom out. And there are others, but this is by far kind of the best studied and the best well-known of the bunch. Well, by kind of constantly pumping sodium against its concentration gradient and keeping a high concentration of sodium in the extracellular fluid versus the intracellular fluid, we get a hill, right? We get a gradient. We get high here, low here. That makes a gradient. Well, guess what happens when we have a hill, right? Stuff moves down it. That sodium pump that we were just looking at is a form of what we call primary active transport. Primary active transport directly uses the energy released from ATP. By using ATP as an energy source, we create the high concentration of sodium outside and the low concentration of sodium inside, and we create that gradient in the sodium concentration, that hill in the sodium concentration. Well, when we have a hill, we have a form of potential energy. And so we have secondary active transport, which doesn't directly use ATP, but indirectly uses it, because what its source of energy is really the potential energy from the concentration gradient. So we had our sodium-potassium pump that pumped sodium out into the extracellular fluid. That was primary active transport. Well, now we have a carrier protein over here that's going to allow sodium to diffuse down its concentration gradient, but bring glucose, something that is high inside the cell but low outside the cell. Glucose gets to kind of come with it. And even though glucose is going against its concentration gradient, it is using the sodium concentration gradient as a form of potential energy. And that allows us to bring very large, chunky molecules into the cell sometimes. So that's secondary active transport, using not ATP directly, but using the concentration gradients as a form of potential energy. The other type of active transport is what is known as vesicular transport. And really what a vesicle is basically a little sack of phospholipids filled with some sort of contents. And we use vesicular transport, particularly if we have really, really large things that we have to move, things like some of our big macromolecules, our big clunky proteins, and nucleic acids and stuff like that. And then also fluids. One of the ways the cells can kind of sample the fluid around them is by using vesicular transport. So since we are using these, we're kind of building these little sacks, that is also going to require energy in the form of ATP. So there are kind of two ways that this can happen, right? So there's exocytosis, which is stuff kind of moving out of the cell, like exo, right? And endocytosis, which is stuff moving into the cell. And then there's actually something called transcytosis, where something will come into a cell, move across it, all right, that's what trans means, and then kind of spit it out on the other side. And so that's transcytosis. The two main forms of endocytosis are phagocytosis and pinocytosis. And phagocytosis is what you're seeing here. So here you have your little cell, and let's say it sees maybe a bacteria, right? It basically extends around the bacteria, sucks it in, and then usually merges it with a lysosome, which has a bunch of digestive enzymes that can kind of eat it, essentially. And then pinocytosis is when the vesicle kind of forms around the fluid in the extracellular environment, and is essentially a way for a cell to kind of sample its extracellular environment and kind of see what the ion concentrations and other types of things are out there around them. If you go back here, let me go back for a second. If you go and click on this video here, you'll be able to see a little video of what exocytosis and endocytosis looks like. So I definitely recommend doing that. The other thing I want to discuss here in part 1 is how cells interact with their environment. And there are really kind of 2 ways that we do that. We either respond, to another cell, or respond to some sort of kind of chemical messenger that's in the body. These cell-to-cell or cell-to-environment interactions are always going to involve that kind of sugar-coating glycocalyx, and so those receptor kind of sugar proteins and sugar lipids that are part of that kind of ID tag glycocalyx. So if we're talking about a direct cell-to-cell interaction, we're usually talking about what we call contact signaling, i.e. you've got a couple cells, they are touching each other, they recognize each other because they all have unique glycocalyx signatures, and then they're able to kind of interact and communicate with each other essentially through touch. This is very, very common in the development, like embryonic development, because that's a process that is really highly controlled for it to occur properly. And also immunity. When one of your immune cells kind of recognizes one of your own body cells, it says, "Oh, okay, you belong to me. I don't have to deal with you." But if it kind of touches something that it doesn't recognize and something that seems foreign to it, it'll be like, "Oh, wait, you don't belong to me. I have to get rid of you." So immunity functions that way too. The other way that we have common kind of interactions between cells and extracellular things is what we call chemical signaling. This is what happens with hormones in the blood, neurotransmitters in the brain and the nervous system. And it's essentially an interaction between a receptor in that glycocalyx on our cell and some sort of ligand or chemical messenger. And essentially what happens is the chemical messenger, the ligand, binds onto the receptor. That receptor then changes shape and something else happens. It triggers some other type of response. And those responses can vary. If the receptor was a catalytic enzyme, it might then become an activated enzyme as opposed to an inactive enzyme when signaled by some chemical messenger. If the receptor was a gated channel, well then, here's our little receptor protein, when the chemical binds onto it, the ligand binds onto it, then the channel opens, and that changes what we call excitability or responsiveness. And then the other kind of major type of receptor that we usually find in our plasma membrane are what we call G-protein linked receptors. And G-protein linked receptors can cause really systemic effects in terms of changing the kind of functionality of a cell. And they work through what we call second messengers. If you think of the initial kind of ligand binding onto the G-protein linked receptor, then second messengers are things that are released inside the cell. You can almost think of it like a relay race. So if you think of the ligand as the first messenger, it kind of hands off that message to the receptor. The receptor is then linked to a G protein, so that receptor hands off the message to the G protein. The G protein then goes and activates some sort of enzyme that's also bound into the plasma membrane, and then the plasma membrane hands off the message to the second receptor or the second messenger, and now that second messenger is free to do kind of different activities inside of the cell. And one of the reasons this process can have such huge effects is, let's start at the beginning and we'll see. So here's our ligand, our first messenger. It was released from someplace else in the body and traveled either through the blood or through the extracellular fluid, and then it binds onto the G protein linked receptor. There's the receptor, there's the G protein. The ligand hands off the message to the receptor, which hands it off to the G protein, which goes and activates this other enzyme. That second enzyme here then activates our second messenger. And it can actually activate lots of these second messengers. And then these second messengers usually activate what we call protein kinase enzymes. And protein kinase enzymes can do all kinds of things. They can cause structural changes in the cell. They can change metabolic rates of the cells. They can open and close ion channels. They can affect protein synthesis, and because we get lots of second messengers being activated, we also get lots and lots and lots of protein kinases being activated, and we get kind of an amplification of the response. And so hormones, for instance, there are certain hormones that cause really huge cascading effects in the body, and they usually work through this G protein linked receptor process.